Doktor Derviş Salih, Alzheimer hastalığı, COVID-19 ve uzun ömürlülük ile ilişkili doğuştan gelen bağışıklık sisteminde ortak genetik varyasyon. Doktor Derviş Salih, lisans eğitimini King's College, Londra Üniversitesi Moleküler Genetik alanında, doktora eğitimini Cambridge Abraham Enstitüsü ve Wolfson Koleji Moleküler Endokrinoloji ve Fizyoloji alanında tamamlamış ve yine aynı alanda Abraham Enstitüsü'nde doktora sonrası çalışmalarını sürdürmüştür. Sonrasında Stanford Üniversitesi Genetik Bölümünde doktora sonrası öğretim üyesi olarak moleküler sinir bilim ve yaşlanma konularında çalışmalarına devam etmiştir. Ardından bir süre Londra College Üniversitesi Sinir Bilim Bölümünde fizyoloji ve farmakoloji alanında araştırma üyesi olarak görev yapan Doktor Salih, şu an Birleşik Krallık Alzheimer Araştırma Merkezi'nde kıdemli araştırma görevlisi olarak Alzheimer hastalığında yaşlanma ve COVID-19 arasında moleküler bağlantıları anlamakla ilgili çalışmalarını sürdürmektedir.
Bioinfocongress II Dr. Derviş Salih Common genetic variation in the innate immune system associated with Alzheimer's disease, COVID-19 and longevity. Dr. Derviş Salih achieved a bachelor's degree in molecular genetics at King's College, University of London, UK. His PhD is in molecular endocrinology and physiology from the Abraham Institute and Wolfson College, University of Cambridge. He then performed research as a postdoctoral fellow in the fields of molecular neuroscience and aging at the Department of Genetics at Stanford University. After a while, he continued his studies as a research associate in molecular neuroscience and electrophysiology at the Department of Neuroscience, Physiology and Pharmacology at University College London, UK. Dr. Salih is now a senior research associate at the UK Dementia Research Institute at University College London, where he continues to investigate the molecular links between Alzheimer's disease, aging, and COVID-19.
Dear Dr. Salih and our valued participants, welcome all of you to our Bioinfocongress II event. Our esteemed teacher will give us a talk today on common genetic variation in the innate immune system associated with Alzheimer's disease, COVID-19, and longevity. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Derviş Salih, welcome back.
Thank you. Thanks very much for this kind opportunity. You'll tell me if you can't hear me or if the slides are not behaving themselves. I'd appreciate that. Thank you very much.
Yes. If you wish, you can start your presentation now.
Okay, great. So I also want to say it's a really fantastic program that you've put together over these last few days. So it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation. I'll just put my laser pointer on and get started. So I'm a senior research associate at University College London, working at the UK Dementia Research Institute. Today, I'm going to talk about common genetic variation in the human population. This is in contrast to rare variants that are also studied quite a lot for disease. And we use common genetic variation to gain new insights into processes such as Alzheimer's disease, COVID-19, and human lifespan longevity. So I'm particularly interested in the mechanisms, the molecular mechanisms that link a series of dementias, neurodegeneration, mainly Alzheimer's disease, with lifespan control or longevity. And as you'll hear from the rest of my slide, our studies have led us to study microglia, which are the innate immune cells of the brain. So microglia help to support the neurons. They provide them with factors to promote their activity, and also they help to clear any debris or damage that results from injury. And so we've been studying how these innate immune cells, microglia, interact with neuron, and we believe that's key to both Alzheimer's and aging. And for our studies, we use data and we perform experiments with a variety of models, so mouse models of disease, primary cells that are isolated and cultured from mouse models, and also more recently, human stem cells that are differentiated to microglia. And together, this work allows us to build gene co-expression networks, so basically looking at changes in gene activity. And we hope in the future that will allow us to predict disease better and also identify new targets for drug discovery. And the overall aim of my group is to be able to boost the beneficial effects of these immune cells, of these microglia, in neurodegenerative processes. So I'm going to say a basic introduction now about Alzheimer's disease. Alzheimer's today affects more than 50 million people worldwide. So it's quite common, and we still have no drugs to slow down development of this disease. Once you're diagnosed with Alzheimer's, you're probably going to die with Alzheimer's. Alzheimer's has three characteristic features. It has extracellular amyloid plaques, intracellular tau tangles, and the third feature is neuroinflammation, and I'll talk about that in the coming slides. This is the amyloid cascades that was initially proposed more than 30 years ago now by John Hardy and his colleagues to try and explain how the different features of Alzheimer's might fit together into a timeline. And this is the model that they proposed, that familial mutations, so these are rare mutations that occur in families, in genes such as the amyloid precursor protein gene or presenilin 1 or 2, can result in elevated levels of A beta. So this is Alzheimer's that passes in families, and this is early onset Alzheimer's. Most of Alzheimer's is sporadic and it's late onset. It starts to develop in when people reach their 70s or 80s, and that's probably due in part to environmental factors, things like concussion, stroke, maybe diet, levels of exercise. And these genetic and environmental factors gradually lead to elevations of amyloid beta in the brain. The amyloid beta forms deposits over time. And then over a longer time period still, this leads to changes in a protein called tau. And these changes include hyperphosphorylation and then conformational changes. And this, again, later on leads to tau tangles within neurons. And it's these tau tangles that actually cause the death of neurons, that cause the neurodegeneration. So aging also plays a role in this process. The older you are, the more likely you are to reach the end stages of neurodegeneration. But we don't understand the mechanisms that link aging with Alzheimer's and neurodegeneration. The other thing to say here is that this is an extremely slow process, and it probably takes something in the order of 20, 30, 40 years to go from elevated A beta to being diagnosed with Alzheimer's, which would happen when approximately 20% of the neurons in a brain structure called the hippocampus have died. So the hippocampus controls learning and memory. And when a certain number of neurons in that structure dies, then people start to show memory problems. And then that's when Alzheimer's is diagnosed. But it's an extremely slow disease that is going on inside people for 20, 30 years before it's actually diagnosed. So that's part of the reason why we can't treat it, because most of the time it's diagnosed quite late in the disease. So most of the disease is diagnosed at this point. So this was the state of play and what we believe to be true about Alzheimer's. for a long time. But then over the last, and this is a very biased model towards neurons, it's very neuron-centric. A beta is produced by neurons, tau is within neurons, it's all about the neurons, it's the neurons that are dying. And then around 10 years ago, sequencing technology improved, sequencing of DNA, and this led to genome-wide association studies. And genome-wide association studies work by looking at differences in DNA sequence between one large population of people with Alzheimer's and a matched population of controls without Alzheimer's disease. And by comparing the DNA sequences, we can get insights into what changes in the DNA sequence increase risk for Alzheimer's. And these were first applied to Alzheimer's around 10 years ago now. And the initial paper in this area identified TREM2 as showing DNA variation associated with risk for Alzheimer's disease. So TREM2 is expressed by the immune cells of the brain, by microglia. So initially it was a surprise and many people didn't believe this, but then over the years, so you can see 2017 and another paper in 2017, more risk genes were identified in addition to confirming TREM2. A Swedish group also identified TREM2 variants as well, Johnson, in 2013, but the other genes that began to be identified as well, PLCG2, ABI3, and the transcription factor PU1, SPI1, also are expressed by the innate immune system, and this was a surprise. And then over the years, around 50 to 70 genes have been identified using GWAS that contribute towards risk for Alzheimer's, and these are mainly expressed by the innate immune system. So despite these findings around 10 years ago, we still don't entirely understand how these immune cells, how microglia contribute to dementia. A number of groups now have been studying how these risk genes for Alzheimer's affect the function of microglia. So these brain sections from mouse models of Alzheimer's disease that contain the familial mutations in APP and presenilin-1. And these mice develop amyloid plaques like in humans. So these are marked in blue. And the green cells here are the microglia, they're their immune cells. And you can see that these immune cells cluster around the amyloid beta pathology. When some of the risk genes associated with Alzheimer's are mutated, so here we're looking at a TREM2 knockout, or here knocking in mutations into TREM2, we see that the immune cells can no longer migrate to the border of the plaque and surround it. And so it seems like part of the role of microglia in Alzheimer's disease is to surround the plaque, and the hypothesis would be either to contain the damage, but there are also studies that show that these microglia can also make the pathology worse by spreading the amyloid beta. Another intense area of study is the phagocytosis by microglia. Microglia have the ability to engulf this material, and it's been proposed that microglia may be engulfing the amyloid beta, trying to remove this damage from the brain. Other groups, such as Soyeon Han, also at UCL, have shown that microglia can phagocytose synapses, and if this happens too aggressively, then you're losing too many synapses from the brain, and that may also make the dementia worse. Also, other groups have shown that microglia can actually modify the structure of the amyloid beta plaque. So here the plaques, we can see they're quite dense, they're quite compact, and that might be a way by which the microglia are limiting the damage to a small area. In other studies where you mutate the risk genes associated with Alzheimer's, you get more diffuse plaques where the amyloid beta spreads in a more random fashion. It almost looks like a star shape, and that may cause more damage to synapses and neurons. And so really, there's still a lot that we need to understand about how these immune cells contribute to dementia. The other interesting point is at what stage of this amyloid cascade are these innate immune cells functioning? And so you can see that this is a multi-step process that takes place over several decades. And I believe it would be useful to know at what stage these risk genes for Alzheimer's are involved, what stages they're regulating. And the obvious places to put them would be in response to amyloid beta or in response to tau tangles, so regulating this red stage or this blue stage. And so that was one of the first studies that we performed in this area to gain better insights into what genetics was telling us about Alzheimer's disease and the innate immune system. And so the hypothesis going into these experiments was that genetic variability in the human population in response to cellular damage influences risk of disease and also lifespan. And in the context of this work and this talk, when we talk about cellular damage, we're talking about damage resulting from amyloid beta or damage that occurs with aging. And as we were performing these experiments, the types of questions we were thinking about were, can we understand what links together these risk genes that have been identified like TREM2 and SPI1? If we can understand these links and mechanisms better, can we use that to predict new risk genes for Alzheimer's and also for lifespan? These GWAS data, they've been going on for around 10 years now. The modern GWAS have hundreds of thousands of people's worth of data in there. So they're very expensive studies and they take a long time. Can we make better use of this GWAS data? Can we get new insights? And then with these insights, can we make new progress into understanding mechanisms involved in Alzheimer's and aging? So these are the things that we're thinking about. And the general approach that I use for my work is to integrate different data sets together to get new information. So we use data from Genome-Wide Association studies, GWAS, as I've mentioned before, at the population level. And we filter this data with transcriptional changes that are associated with disease and lifespan in human tissue and also in mouse models. And together, this gives us new insights that will in the future allow us to predict disease risk better, but also provides new ideas for drugs where we can intervene in this process to slow this process down. And so today, using this approach and this background, I'm going to tell you three brief stories. The first of this is identifying new genes associated with Alzheimer's disease. Then I'm going to talk about genetic risk of critical illness with COVID-19. And then finally, I'm going to talk about genetic variation associated with lifespan, with longevity, in genes that are expressed by the brain cells. And so a big part of our studies is making better use of these rich genome-wide association data sets. This is a Manhattan plot here. Each of the spots here is a change or a difference in the DNA sequence. And so you can see all of the chromosomes are represented here on the x-axis. And the variants that we're still looking at, that most people are still looking at, are single nucleotide polymorphisms, SNPs. And on the y-axis here, we have a p-value for the significance of association of these DNA changes with whatever process you're interested in. Here we're looking at Alzheimer's disease. So each one of these SNPs, the height on the y-axis, is telling us about association of that SNP with Alzheimer's disease. So given that so many SNPs are sampled across the genome, for a SNP to be significant at the genome-wide level, its p-value needs to be less than 10 to the minus 8, so it needs to be above this dotted line. And you can see that there are a few SNPs here which are significant genome-wide, and they represent the genes that I've already presented, such as ApoE and TREM2. You can see that there's also a number of SNPs that are within these two dotted lines. This lower dotted line is 10 to the minus 2. This is the type of nominal p-value that a biologist would use in a hypothesis-driven experiment at the bench, so in the wet lab. And you can see that a number of SNPs here, while they're not significant at the genome-wide level, they still may give us new insights into disease. The other thing to say here is that these GWAS studies, they still have limitations. And these are manifold. Two of the most obvious limitations are that SNPs may be in different positions in different people, and also these SNPs might be different in different populations, so Europeans versus Asians versus Africans. And most GWAS studies are still performed on European populations. And so if these SNPs are different in different individuals, then these p-values kind of drop, and it brings them into this sub-threshold range that we're looking at. And the other obvious limitation is that some of these DNA changes, they're not just involved in Alzheimer's, they're involved in other processes, other diseases such as diabetes, maybe multiple sclerosis. And so if a gene is involved, if a SNP is involved in multiple processes, its effects are going to get diluted in these experiments, and it's going to drop below this top dotted line. And so basically, I'm interested in these sub-threshold hits. We want to try and mine that data to get new insights. And so the hypothesis here becomes that it's not these few SNPs above the dotted lines that control your risk for Alzheimer's. In fact, it's a combination of all of these SNPs with small effect sizes that contribute towards dementia. And so that's what we're studying here. So one way to make better use of this GWAS data is to summarize or combine together the statistics for several SNPs across each gene. So here we're zooming into the Manhattan plot. This is an example of one gene that we've been studying, OAS1, oligoadenylate synthetase. I'll say more about that gene later on, but here I'm just illustrating the principle. Here we're looking at the first exon of the gene to the last exon. And you can see from the beginning to the end of the gene, there are multiple SNPs that we can survey for that gene. And here again, we have the nominal p-value line plotted as a dot here, as a dotted line. And what we're doing is we're summarizing the statistical information for all of these SNPs. And for this particular gene, you can see that a number of the SNPs are above the nominal p-value of significance. So this way, we collapse the statistical information for all of the SNPs into one p-value for each gene in the genome. And that's what we're using in the studies. So now I'm going to say how we use this information to get new genetic ideas into what's controlling risk for Alzheimer's disease. And so to combine the GWAS data with the transcriptional filter, we need to assess transcriptional changes with Alzheimer's disease. And for this, we used a series of transgenic models for Alzheimer's disease. So these mice have either amyloid beta pathology. So these are the star shapes you can see here in green. These are all brain sections from mice with amyloid pathology at varying rates. And in the top panels here, mouse models which have tau pathology. So here you can see the green signal is representing hyperphosphorylated tau, and this occurs within neurons. So we're using transgenic mice from 5 lines, and these carry the familial mutations that are found in early onset Alzheimer's disease. So mutations such as M146V in presenilin-1, mutations, familial mutations in APP, and then mice which have 1 copy of each gene, or homozygous 2 copies of each gene, 2 copies of the mutations in APP and presenilin-1. And you can see that there's a gene dosage effect here. So if you have more of these Alzheimer's inducing genes, then you get pathology much sooner, and it's more extensive. Along the x-axis here, we've got the age of the animal, so going from 2 months of age to 18 months of age. So these most extreme line of the transgenic animals begin to develop pathology around 4 months of age. And these tau transgenic animals start to show pathology from 8 months onwards, or actually before 8 months. It's quite established at 8 months already. So these levels of pathology, the amyloid pathology and the tau pathology, we can quantify in brain sections, and then we can compare that to gene expression changes, gene activity changes by RNA sequencing. And I also want to say that all of our RNA sequencing data is available on this website, mouseyak.org. So later on, you can go in and look for your favorite genes to see how they're different, how their activity levels are different in the different mouse models. Here we're looking at one of our favorite genes, TREM2. And the black line here are controlled mice. They're wild type mice, black 6 animals. From 2 to 18 months of age, the gene activity levels of TREM2, it doesn't change very much. These red lines here are representing the 2 Alzheimer's mouse models with the more extreme pathology. And you can see that TREM2 activity levels increase from around 4 months of age onwards. And so this correlates to the changes in pathology, to the amyloid pathology in these mice. And so RNA sequencing, it gives us a lot of information. We can detect reliable expression for around 18,000 genes in the mouse brain. And that's a lot of data to process and to understand. And to make better use of that data also, we use a lot of bioinformatics approaches. And what's been really useful for us is an approach called gene co-expression network analysis, or WGCNA. Some of you might be familiar with this already. What this does is it actually goes through all of the gene expression patterns that we can detect, and it finds genes which have a similar expression pattern. So this is the expression pattern that we were looking at for TREM2 in the slide before. These are other risk genes for Alzheimer's, ABI3, MS4A6D. These also have similar expression patterns, you can see. And this also correlates very tightly with the levels of amyloid pathology in these mice. So using this bioinformatic tool, we pull out all of the genes which have this expression pattern. And altogether, there's around 1,500 genes which have this expression profile. This shows these genes together, their expression, have a very tight correlation with the amyloid pathology. It's 0.94. You can see that the p-value is really low, e⁻41. And so basically, there's a very close link between the activity of these genes in this network with the amyloid pathology. And here we're looking at the top 150 genes of the 1,500. And each circle here is a gene. And if there's a green line between 2 genes, it means that their correlation between their expression levels is above a certain threshold, their correlation or their connectivity. And the genes in the center here are what we call hub genes. They have the highest correlations to the rest of the network. You can see here that TREM2, I've marked it with this red circle, this red oval. It's sitting in the center of this network, and these hub genes are thought to be driving the response to amyloid pathology. So most of these genes are actually expressed by the microglia, by these immune cells, and how they respond to amyloid. So we're getting all of these clues coming together now, and we find that this network of genes is actually statistically enriched for risk genes associated with Alzheimer's disease from the GWAS. And everything that's marked with the red oval here is a known risk gene for Alzheimer's. So ABI3 we've seen already, SPI1, MS4A6D, CD33, they're all known risk genes for Alzheimer's. And what we're doing now is that in the human genome, there's around 40,000 genes with RNA-seq. We can measure 18,000 genes. We're reducing down the number of genes that we're interested in to 1,500 genes. And by lowering the number of genes we're looking at, the subthreshold genes that we were looking at in the Manhattan plot now become significant because we're doing a more focused statistical analysis. And that gives us new risk genes for Alzheimer's, and these are marked with the blue rectangles here. So these genes now become significant when we reduce the sample, the number of genes that we're looking at. And these new risk genes that we've identified are ITSGAM, also known as CD11B. This is a marker for microglial cells. We identify this lysosomal protein, LAPTM5. Here, LILRA5 is a leukocyte receptor. And this gene here that you've heard about already, OAS1, is actually an antiviral gene. And you can see its expression levels, its activity patterns are very similar to TREM2 and the pathology. So this was work that we published at the end of 2019, and basically we have around 15 more risk genes that we identified, and those are listed here. I'm not going to go through all of them, but you can have a quick look, and if any of them seem familiar to you, we can talk about those afterwards. Okay. So basically now we're coming to the model where it's not a limited number of genes like TREM2 and ApoE. that are controlling risk for Alzheimer's disease. It's actually a combination of lots of genes working together. Some people have good mutations in these genes, some people have bad mutations, and together those genes work together to control the risk for Alzheimer's disease. And other studies that I don't have time to show today, we've shown basically that the risk genes associated with Alzheimer's from the GWAS are regulating this red step here. They're responding to amyloid beta, and they're controlling how amyloid beta converts to tau pathology. When we do a similar analysis in the tau transgenic animals, the risk genes that we identify, the gene network that we identify associated with tau is not enriched for risk genes associated with Alzheimer's. And so the gene expression changes in microglia are less likely to be regulating this step here. And so that gives us new insights into how these innate immune cells are contributing to Alzheimer's disease by responding more specifically to amyloid beta pathology. And so we also approach the model whereby combining together all of these variants is telling us that if you have a bad collection of mutations to your microglial genes, then you're going to move down this amyloid cascade really quickly and start developing neurodegeneration in your 70s or 80s. If you have a good collection of SNPs, then you're going to move down this amyloid cascade more slowly. And even at the age of 90, 100, then you might have some amyloid beta in your brain or even some tau tangles, but you're not going to have cell death, you're not going to have neuronal death, and you're not going to show the cognitive problems associated with Alzheimer's disease. So that's the type of model that our data is supporting. So now I'm going to switch gears slightly and talk about genetic risk associated with COVID-19. And it's a good question to understand what the links are between Alzheimer's and COVID-19. We know that COVID-19 seems to affect older people more severely. So obviously, COVID has been really bad for the world, and it's changed our lives, and it will change our lives in the future as well. But science has really given us a silver lining. Science has moved really quickly. And last year, a Genome-Wide association study came out for severe COVID-19. So these are people that have a really bad response to the COVID-19. They need to go into hospital and they need to go into critical care. So they need manual automatic ventilation to help them cope with the COVID and to enable them to breathe. And this group in Edinburgh, they identified these 10 genes which increase risk for severe COVID-19. So these genes are all expressed by the interferon responsive pathway. And we were surprised when we saw this data because 3 of the genes that I've marked in red here were present in our amyloid responsive network in the transgenic mice that were responding to amyloid pathology, again, expressed by the innate immune cells. And OAS1 was one of those genes. And so we became really interested in understanding how variants, DNA changes in OAS1 were altering risk for both Alzheimer's and COVID-19. And what we did was we mapped the position of these DNA variants. So these are the SNPs here. SNPs are given these numbers following RS. And the 2 red SNPs here are the SNPs that we found to be associated with Alzheimer's in our study that I've just discussed. And the blue SNPs here were those found by the Edinburgh group to be associated with critical illness for COVID-19. And you can see basically that these SNPs are overlapping with one another, and they're marking the genomic location between the OAS1 gene and the OAS3 gene and the region in between. And with further analyses within our collaborators, Kevin Morgan and Mariam Shoa, we see that these variants are significantly linked. And so people that inherit a bad copy of this genetic variation are likely to be inheriting bad copies of these other SNPs as well, because they're so close together in the genome, they're inherited together. And what we think these variants are doing are actually reducing the expression levels of OAS1. So they're making the OAS1 less active. And that means these SNPs are acting as what's called an expression QTL, quantitative trait locus. So expression quantitative trait locus. If we look at other data sets, such as the 1,000 genomes data set, this is mainly looking at the European population. We see that having this particular combination of DNA sequence, these 4 SNPs, so these are the 4 genomic loci, ATTA, having that particular combination would increase risk for both Alzheimer's and COVID-19. And you can see the frequency of this particular combination of DNA sequence is actually quite high in the European population. It's extremely common. 56% of people have this particular combination of variants, which may form a haplotype. And so this might explain why so many people respond strongly to the SARS virus for COVID-19, and also why Alzheimer's is quite common in the population. So about a third of people at the age of 85 years of age have Alzheimer's disease. So that's also quite common. This particular combination of DNA sequences, G-CCG, is actually the protective version of these. And that's present in about 35% of the population. So you can see that it actually is affecting quite a lot of people here. So OAS1, what is it doing? It's an antiviral enzyme traditionally. It's called oligoadenylate synthetase. What it was known to do was to respond to double-stranded RNA within viruses. It binds to the OAS1. It causes conformational changes within OAS1 to make it active. The OAS1 then produces oligoadenylate, which activates these intracellular ribonucleases. Ribonucleases, what they do is they act like molecular scissors to chop up the RNA and to make the viral RNA inactive. And so what OAS1 and ribonuclease are doing is that they're protecting the cell from viral RNA. And this process of recognizing and cleaving the RNA produces more interferons, and then this causes a feed-forward loop where you get further interferon signaling through interferon receptors, activating the JAK-STAT pathway to cause further activity increases in OAS1 and other interferon-responsive genes. So this process is very well understood for the innate immune system and how it deals with viruses. However, the question is, how is this related to Alzheimer's disease? And we still can't understand that. We still don't understand that question very well. But the hypotheses that have been proposed, that this system is responding to nucleic acids, to RNA, and also maybe certain structures of DNA that are produced when neurons are dying. They may release their nucleic acids. Mitochondria also have DNA. If cells are distressed, if they're under stress, then they may also release their mitochondrial RNA and DNA to activate this process. Also, amyloid beta, it acts as a very rigid and strong net to trap nucleic acids. And it's possible that this pathway is also responding to the nucleic acids that get trapped within amyloid beta plaques. So these are the types of ideas that we're thinking about in our experiments. And also what's missing is an understanding of the genetic network that controls risk for both Alzheimer's and COVID-19. And so to get more insights into that, we performed these gene activity studies again, these pathway analyses. And we identified this genetic network. This time we're using single cell RNA-seq, so isolating microglia from mouse models of Alzheimer's disease, this time an APP knock-in mouse with a control mouse, a wild-type mouse. And this gives us this genetic network, which contains OAS1. So OAS1A is the mouse version of the human gene, which is OAS1. And this particular network is expressed by interferon responsive microglia. And this is illustrated by this trajectory plot here, or pathway plot. Each one of these dots here is a single microglial cell, and where the dot is for each cell is summarized by the expression of all of the genes within the cell. And this is put onto a two-dimensional plane according to the Y and the X axis. And so this way, the different types of microglia within the brains of these animals separate into different subpopulations. So here on the left, we have a homeostatic or a resting population of immune cells. On the right here, we have amyloid responsive microglia. And at the top here, we have interferon responsive microglia. So this key here shows that this combination of genes is expressed most in the yellow and orange cells, which are the interferon responsive microglia. These collection of genes are increased in expression in old animals, 16 to 18 months of age, compared to young mice, 6 to 8 weeks of age. In a separate analysis of data from a different mouse model of Alzheimer's disease, this collection of genes again shows increased activity in mice with amyloid compared to control or wild type animals that don't have amyloid pathology. So these genes within these interferon responsive microglia are responding to age and to amyloid pathology. So this is mouse data. We can do the same with human data. So now we're looking at single cell RNA sequencing in immune microglia, immune cell microglia, isolated this time from people's brains. So this is pathology tissue from people with Alzheimer's disease and mild cognitive impairment. So this is before they have the later severe stages of Alzheimer's disease. And when we do the network analysis here, you can see we derive a network which is very similar to the mouse network I showed before. It has OAS1 and many other interferon responsive genes. This is again expressed by the interferon responsive microglia. And here on the right, we're looking at how the expression of these genes, how the activity of these genes changes with age in the human population. And you can see that these genes are also responding to aging because their activity increases with age towards the end of life. So this is microglia from people with Alzheimer's disease. We can also do the same with pathology tissue from people with severe COVID-19. So this time we're using single cell RNA sequencing from lung macrophages. So these cells are the lung equivalent of the brain immune cells. And looking at these macrophages from people with severe COVID-19 compared to people that have a healthy response to COVID-19, so they don't get a very strong version of the disease, they can recover at home, versus healthy individuals, we derive this network, which although it contains more genes, it actually has a very similar structure. Again, it has OAS1, which is quite close to the hub. It has this ISG15 gene in there again, and IFIT-M3. And we find that this particular combination of genes expressed by these lung macrophages show increased activity in people with a strong response to COVID compared to people with a mild response versus healthy controls. So this particular combination of genes is controlling how these lung macrophages respond to the COVID-19 virus. And so actually it's quite surprising that the genetic networks that we identified, this is the same data again. This is the gene expression network from the microglia in the Alzheimer's brains. And this is the network from the lung macrophages in severe COVID-19. And we can see that the genes present in these networks show a significant overlap. So e to the -21, both contain OAS1, both have ISG15 driving the response of these cells. And so what this is telling us is that these innate immune cells, whether they're in the brain or the lung, they're responding similarly to the amyloid pathology and to the SARS virus for COVID-19. So we believe that there's a lot of data and information here that we still need to understand better. For example, are there good targets in these networks to allow us to develop new drugs to try and slow down the processes in Alzheimer's or to give a milder response in COVID-19? Also, a number of these genes here, such as ISG15 and some of the CCL molecules within these networks, they're actually secreted outside of the cell. So if we measure the levels of those proteins in the blood, in the serum, or in the cerebrospinal fluid in the brain, then can we use that to predict why some people have strong versions of these diseases and why other people have milder versions? So basically, can we track the progression of disease better by identifying some markers within these networks? So those are all experiments that we're thinking about. We wanted to understand more at the cellular and molecular level what this antiviral OAS1 gene was doing. And for that, we cultured human stem cells that were differentiated to microglia in collaboration with Jennifer Pocock's lab at UCL. And we manipulated OAS1 activity levels to try and mimic what the genetic data was telling us. The genetic data, to remind you, it was telling us that OAS1 levels were decreased. There was reduced production of OAS1 mRNA. And so we modeled that using RNAi interference. And this plot here shows basically that with the OAS1 knockdown siRNA, we can reliably reduce the expression levels of OAS1. So the y-axis here is the expression level of OAS1, and we get significant knockdown of OAS1 under basal conditions, and also in response to interferon gamma stimulation, which is an important component of the response to the SARS virus and COVID. And so we can knock down the activity of OAS1 in these cells. These stem cell-derived microglia, generally with reduced OAS1 activity, they behave quite normally. Their levels of homeostatic genes are normal. Their levels of phagocytic genes are normal. Where we do see a difference is in the production of pro-inflammatory cytokines, such as TNF-alpha. We find that in response to interferon gamma, reducing OAS1 expression levels gives an exaggerated production of these pro-inflammatory cytokines, such as TNF-alpha. So this is at the mRNA level. We get an exaggerated production of mRNA for TNF. And also at the protein level, this is an ELISA now, measuring TNF-alpha levels in the media, so secretion of TNF-alpha into the media. This is also increased when OAS1 levels are knocked down with the RNAi interference in response to interferon gamma. And so the model that this data is suggesting is that OAS1, in addition to controlling antiviral activity via the interferon response, it's also suppressing, under normal conditions in people with healthy variants of OAS1, it's suppressing the pro-inflammatory response. Pro-inflammation is bad because if it's chronic, if it's continued for a long time, it can begin to damage and kill neurons in the brain. And also in COVID, other clinical labs have shown that pro-inflammatory, over-exaggerated pro-inflammatory stimulation, so what people are calling a cytokine storm, is actually damaging the lung tissue and leading to severe COVID. And so if you have bad mutations in OAS1, this brake is released and you get more pro-inflammatory signals, which lead to dementia via genes like TNF-alpha, or also would lead to severe COVID in lung tissue. We also know that pro-inflammatory stimuli also are suppressing protective signals such as TREM2. And so OAS1 may be controlling multiple pathways towards dementia. And this work was published towards the end of last year in the journal Brain. So there's more information here if you're interested. And so in the final part of my talk, I'm going to talk about genetic variation associated with lifespan, with human longevity in genes that are expressed by the brain. And so this is an ongoing study that's not complete yet, but I'll describe our process so far. What we're doing is basically the same approach using these genome-wide association studies. This time we're using data from these two papers from Deline and colleagues and Timmers. And here they're looking at genetic variation associated with 3 different measures of human lifespan. So the first group of data they're looking at is longevity, genetic variation associated with somebody surviving to the 90th percentile of life or the 99th percentile of life compared to the 60th percentile. So the 60th percentile is what they're considering to be a control lifespan, and basically looking at what genetic variation is driving the difference between living a long time versus living to something that's close to average, the 60th percentile. Another measure they're looking at in these papers is parental lifespan. So in families, if you have a relative, a parent, which lives a long time to the age of 90 or 100, then you are also more likely to live longer. And so they're looking at genetic variation between families, between parents and their children, to see if they can understand what genetic variation is associated with lifespan. And the third and final measure that they're looking at is health span. So people surviving to a long age without obvious signs of disease. So people that don't get the normal age-dependent diseases such as diabetes, Alzheimer's, cardiovascular disease. So what genetic differences, what genetic variation, what SNPs are driving health span. And so we reanalyzed all of this data, again, using that approach to summarize the p-value for all of the SNPs, for all of the single nucleotide polymorphisms within the boundaries, the beginning and ends of genes, to give us gene level p-values. So that's what's shown here. And this was the approach developed by my collaborator, Valentina Escott-Price, also at the UK Dementia Research Institute at Cardiff University. And what we're looking at here are the p-values for these genes. So these are the gene level p-values that contribute to survival to the 90th percentile of life versus the 60th percentile of life. So to put this into perspective, a male living to 89 years of age is the 90th percentile of life, and a male living to 75 years of age is the 60th percentile of life. So the 14-year gap here is contributed to by variation in these genes. And depending on if you're an optimistic or a pessimistic person, it's probably not a surprise that ApoE is the top hit here, e to the minus 35 p-value. And a number of other genes, if we look down this list, we find that they're expressed by the innate immune system, by microglia and related cells. And so we began to get really interested in this data and understanding what this data was telling us. If we keep working down this list, you can find that some genes here are also relevant to Alzheimer's disease. So this is APP, the amyloid precursor protein gene, which also has variation associated with lifespan. And I should say that the genetic variation that's identified in this study are not the rare mutations that you find in Alzheimer's that give early onset Alzheimer's. These are common variants in APP that are somehow contributing towards lifespan. And so these p-values are more modest. They're e to the minus 5. And these e to the minus 5 genes are basically those sub-threshold genes that we want to mine better. And you'll guess exactly what we've done. We've used the same approach here. We've used this network analysis approach, this WGCNA, to look for changes in gene activity. So here, what I'm representing is changes in gene activity in control wild-type animals, black 6 animals, from 2 to 18 months of age. Here, it's the same data, but this time just the controls. And you can see that the activity of these genes increases between 2 to 18 months of age. So here we have the lysosomal protein again, LAPTM5, and then other genes which show a similar expression pattern. And again, that helps us derive these co-expression networks. These are the networks that are expressed by mouse microglia in the brain. And this genetic network on the top here is from human microglia, so from human brains. Both of these networks are from mice or people without Alzheimer's pathology. So these are what are considered healthy organisms that are aging into life. And yet we pull out these microglial networks, which look very similar between human and mouse. And also they're very similar in aging and also in the Alzheimer's networks that I showed earlier. So a number of the genes, ApoE, LAPTM5, TREM2, they're present within these networks. And when we apply this network analysis to the human data, Grace 60 is what we're calling this module, the collection of genes. We find a number of modules. Modules are collections of genes, are networks of genes, which are significantly associated with aging. And so now we're basically narrowing down the data, we're filtering the data to help us understand which genes are more important than others. And we basically have different networks of genes that then we can go and use to test statistically for genetic variation from the GWAS. So here we're looking at 10 different gene networks. Some of them are from human, some of them are from mouse, some of them are from bulk RNA sequencing, some of them are from single cell RNA sequencing of isolated microglia. And what we're looking at is enrichment of genetic variation from the human GWAS with the collections of genes in these genetic networks. And if we look at the Alzheimer's, at the AD GWAS, we can see that the genetic variation from this study is enriched in genes that are expressed by human microglia. So this is not a surprise. The surprise here is that these microglia were identified from older people, not from people with Alzheimer's disease. So again, what this is telling us is some of the processes that take place in aging microglia are actually similar to the processes that take place with normal aging as well. What was more of a surprise was that genes expressed by oligodendrocytes in the brain. So the oligodendrocytes, you may have heard yesterday, are the myelinating cells of the brain. So they produce the myelin that surrounds the axons and helps the transmission of signals between neurons. The genes expressed by oligodendrocytes also encode some of the risk for Alzheimer's disease. So the p-value is not as strong for the microglia, but there's still an enrichment of variation associated with Alzheimer's enriched in microglia and in oligodendrocytes. And then finally, the other thing I want to say here is that this is not quite significant. This is just a tendency. But if we look at the single cell networks from mouse microglia, we can see that there's a tendency and for enrichment of variation associated with Alzheimer's in amyloid responsive microglia and microglia transitioning towards an amyloid responsive state. So this is all about Alzheimer's risk. Now, if we look at the aging data from the Timmers paper, this is the aging GWAS genetic variation data. We see that overall, there's not much enrichment of variation associated with aging in these networks. The only place where there is significant enrichment is in genes expressed by homeostatic microglia isolated by single cell RNA-seq. And so the homeostatic microglia are the resting microglia that are surveying the health of the brain. They're sampling the healthy brain to try and understand what those cells need to respond to. We're going to spend a couple of minutes now looking at these networks in more detail. So again, we have these trajectory plots summarizing the expression of a single microglial cell, and that way We can group them into homeostatic, amyloid responsive, or interferon responsive microglia. This network that we're looking at here has a tendency to be enriched in risk associated with Alzheimer's, and it has the types of genes we would expect. It has ApoE here, it has antigen-presenting cells, and it's expressed by these amyloid-responsive microglia. And if we look at a different data set, so these mouse microglia isolated from old mice, 16 to 18 months of age, compared to young mice, 6 to 8 weeks of age. And we can see that these amyloid-responsive genes in these amyloid-responsive microglia show significant increases in activity in the old mice compared to the young mice. So again, we're looking at this age dependency of these genes associated with Alzheimer's risk. This network is the homeostatic genetic network in resting microglia. This network is enriched for genes associated with lifespan from the GWAS. This network looks like a homeostatic network in microglia. It's expressing many of the homeostatic markers, known homeostatic markers like TMEM119, P2RY12. But there are some surprises in here. The lysosomal protein, LAPTM5, is here. That's associated with Alzheimer's risk as well. ABI3 is an Alzheimer's risk gene. That's also here. And here we have an interferon receptor as well. And these genes are expressed by these homeostatic cells. So you can see the yellow and the orange signal is expressed more highly in the homeostatic cells compared to the amyloid responsive and the interferon responsive. And here, if we look at the old microglia from mice versus the young microglia from mice, that this network is actually getting downregulated. So these genes are showing decreased activity in old compared to young mice. And what I've said is that genetic risk associated with lifespan in humans is enriched in this homeostatic network. And also in oligodendrocytes, we saw that there was enriched risk for variation associated with Alzheimer's. And so we wanted to look more at the relationship between microglia and oligodendrocytes. And so we're looking at this other data set that was published by Nugent, where basically they're taking young control mice and they're using this demyelination agent, cuprizone, for 5 to 12 weeks of age. And this causes demyelination. And what we're doing in this data here is comparing the gene expression changes in the amyloid responsive microglia and the homeostatic microglia that we saw with aging, with the gene expression changes that take place during demyelination. And so the amyloid responsive genes were increasing with age in control mice, and here they're increasing with demyelination. The homeostatic genes were decreasing with age in this data, and here they're decreasing with demyelination. So these gene networks within these microglia are responding similarly to Alzheimer's, to aging, and also to demyelination. And so we think part of what's happening in aging is changes to the myelin status, which the microglia are responding to. So just some other supporting evidence. We've also performed the association of DNA changes with lifespan using a completely different approach, which is TWAS. This is a transcriptional whole association study, whole genome association study. And again, what this data is telling us is that variation in these genes marked in red here is associated with lifespan. And here we're looking at brain cerebellum tissue from humans as an example. We have other data sets that we're looking at. What this is telling us is that genetic variation in this caspase-8 gene, which some of you may have heard about because it's controlling cell death, it's involved in apoptosis and necroptosis. Genetic variation in caspase-8 is associated with lifespan in humans. So lower expression of caspase-8 is associated with lifespan. When we look at the data in this way, we also get information on other genes that we know to be involved in neurodegeneration, which was also a surprise. But again, it's giving us more clues as to what these genes and what these variants are doing in aging and also in Alzheimer's. MAPT is the gene encoding tau, and also variation in this. Again, this is the common genetic variation, not the familial mutations, the rare familial mutations found in tau. These common variants are reducing the expression of tau, and that's associated with increased human lifespan. And here, variation in the Huntington gene associated with Huntington's disease, another neurodegenerative disease, also shows that variation within this locus is affecting the expression of this well-known Huntington gene, and decreased expression of this is associated with human lifespan. And so I realize I've given you a lot of information here, a lot of raw data to try and bring all of this together now. Our model is that variants associated with Alzheimer's disease are regulating the activation of microglia in response to amyloid. These Alzheimer's associated variants are also controlling oligodendrocyte function. The opposite end of the life cycle of a microglia is its homeostatic state. And we find that variants associated with human lifespan, genes such as caspase-8 and also the transcription factor that you may have heard of, FOXO3, are regulating the expression of homeostatic genes in microglia. And so basically we have a balance here whereby variants associated with lifespan and aging are controlling the different extremes of the life cycle of a microglia, from its homeostatic resting state towards its activated state as it responds to amyloid and changes in myelin. And so this is the model that we're continuing to study in my lab right now. And so to summarize all of the information that I've told you today, I've said that risk for Alzheimer's disease is enriched in genes expressed by microglia, and also our new data suggests in genes expressed by oligodendrocytes, the myelinating cells. I've shown that there's a genetic link between Alzheimer's and severe COVID-19 via this OAS1 gene, and part of the mechanism by which OAS1 functions may be by suppressing pro-inflammatory signals. And finally, I've shown that genetic variation associated with human lifespan is enriched in genes expressed by homeostatic microglia. So thank you very much for your attention. I'd like to thank my mentors, John Hardy, Valentina Escott-Price. I'd like to thank the useful discussions we've had with the Drug Discovery Institute at UCL, Paul Whiting and Lorenzo Magno, the people that we work with there. I'd like to thank Jennifer Pocock and Thomas Pears for their help with the stem cell microglial experiments. And I'd like to really thank my lab for the fantastic work that they did during the pandemic, particularly Nadia and Andy and Kelly, which led the OAS1 work, and Andy, Umran, and Meral, which led the longevity study. And I'd like to thank our sponsors as well. So thank you very much for your attention. I'm happy to take questions.
Thank you very much, sir. If you wish, we can go to questions and answers section prepared by our team and consisting of questions from our participants.
Yes, please. Thank you.
Does COVID-19 trigger cognitive decline in Alzheimer's patients of dementia in infected patients?
So those are longitudinal studies, and I know that lots of groups are studying those, including some of our collaborators at UCL. I think it's still early to say. Probably that is going to be the case. That's the hypothesis, but the data is still being generated. So I think it's too early to say.
Okay, thank you. And our next question is, how much of the pathophysiological mechanisms linking Alzheimer's disease and COVID-19 have been elucidated?
Again, this is very early stages. COVID has only really been around for a couple of years now. In terms of the mechanisms linking Alzheimer's and COVID-19, There's still only a few labs around the world that are studying it. We're studying it. Also, Tony Weiss-Corey is doing some really nice work at Stanford as well, looking at, again, RNA-seq changes in people with COVID-19. And he's also seeing that there's quite a lot of changes to the blood-brain barrier and the immune system that's associated with protecting the brain in people with COVID-19. So again, those are the directions that I think research is going to take. Also, more people are becoming interested in the role of interferon in Alzheimer's disease as well. And a few groups are beginning to study that as well, such as Matt Blurton-Jones at UC Irvine and also Alison Goat in New York are also showing that interferon plays a role in Alzheimer's related processes, but it's really early stages.
Thank you for the answer, Professor. Our next question is, in the interrelationship of immunity and aging process, what are your predictions for possible future viral infections in Alzheimer's patients? How can we protect these patients?
Yeah, that's a really good question, and I'm really interested in that. There is a very controversial area of study in Alzheimer's, which says that viruses are actually seeding Alzheimer's disease. And several groups have been studying that, particularly Rudy Tanzi's group at Harvard. These are really difficult questions to answer because ultimately what you need to do is make a really careful record of a person's infection history throughout their age and somehow correlate that to Alzheimer's. And so those are really difficult studies to do. And so we're really not there yet. I think this is going to be an intense area of study. As I mentioned before, I'm quite favoring more a hypothesis where response to viruses and other pathogens are actually modifying the blood-brain barrier. And then over time, over the course of a person's life, changes to that blood-brain barrier and also epigenetic changes to the innate immune system may predispose some people with a bad combination of variants to increased risk from dementia. But really, I think we're in the early stages of studying these. So I think a lot of work needs to be done before I can answer that properly.
Thank you so much, Answer. And our next question is coming from our participants. It says, since TREM2 mediated signaling functions via proteolytical cleavage of the extracellular subunit, have you also observed any change in the expression states in such metalloproteinase enzymes?
Yeah, that's a really good point. So you've probably gathered from my talk, our work is more biased towards the DNA level and the RNA level. And what we're really missing are changes at the protein level. So other people are studying proteomics, and we also want to do some of that in the future. At the RNA level, we don't see changes in those proteases. And so I can't really say if the expression levels of those proteases are changed very much. Those types of enzymes are generally regulated at the protein level, but it's quite possible, and data from others has shown that the proteolysis of TREM2 is also important for disease. And so, again, I don't have any data for that, but it's an intense area of investigation for other labs.
Thank you so much. And our next question is, how does hyperinflammation, which may be a common factor in COVID-19 and Alzheimer's patients' abilities, affect longevity?
So in terms of affecting longevity, it's still not entirely clear. I guess it's more widely accepted now that chronic pro-inflammatory factors are going to contribute to both COVID, severe COVID, and Alzheimer's by damaging nerves or lung tissue. In terms of longevity, it's not really clear yet. I haven't seen really convincing evidence. I think, again, the model would be that gradual changes, chronic changes in inflammation over time are going to be more likely to cause damage to neurons and maybe myelin within the brain. But again, those studies are ongoing, so that's the best I can answer at this point.
Thank you so much. It was our last question. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this informative presentation. I wish you healthy days.
Yeah, thank you very much. Yeah, thank you very much for hosting me, and those were really great questions. So I hope you enjoy the rest of your day. Thank you very much for listening. Take care.